Hi everyone. Thank you, Winnie, for that introduction, and thanks again to everyone for coming to our Cell Science Symposium. I'm excited to share with you some newer work that we're doing, a new project. And so first, hopefully some of you guys were here for Rick's introduction this morning, and you saw the introduction of some of the motivation for what we're trying to do at the Institute. And so overall, we're trying to better understand how cell states and behaviors are defined, and we want to understand how do these ground rules that define these states, how do they change in the context of morphogenesis, disease state, and differentiation. And so our overall goal for this is to generate a data-driven and visual model of the cell, and we'll do this by doing high replicate imaging and try to understand predictive models, or build predictive models and integrate these different structures to see how cells transition between different states. And so the way we approach this complex system is by simplifying it down. And so one of our major assumptions is that cell behaviors are compartmentalized, and that structure, namely structure level at the organelles, can help us infer what cell state that cell is in. And so our approach is to use induced pluripotent stem cells. We do endogenous tagging of organelles using CRISPR-Cas9 and perform high replicate live cell imaging. We use these imaging data sets to generate machine learning and predictive modeling. And so most of the work that most of you probably are familiar with what we've done at the Institute so far is at the structure level, and so I'll give just a really quick background of what we've done at that level before I move into some of our next stages, which are looking more deeper at the genomics level. So what does this look like when we tag endogenous proteins in the cell? So we're interested in looking at different cell states. One of these obvious states would be the mitotic state, and you can see in this labeled H2B line a really nice example of mitosis occurring. And also on the right there in one of our newer dual tag lines, which is tagging the endoplasmic reticulum and the nucleus. So we also can see how these different structures and organelles are repurposed and reutilized when the cell changes from a different cell state. So another example of a state would be a differentiated cell. So we take stem cells, differentiate them into cardiomyocytes, and we can really nicely see with this microtubule line that the mitotic spindle is being formed, and this is labeled with tuba 1b in the stem cells, but that this is repurposed and restructured, reorganized when these cells become cardiomyocytes. And so we're interested in studying what defines those changes, and can we build models that help us predict how things are changing in the cells. So we can look at this at levels of organelles that are expressed in stem cells and how they differ, or how they change as cells differentiate. But there are also some really interesting structures in the cardiomyocyte that are specifically not expressed in the induced pluripotent stem cells. One of these structures is the sarcomere. So this is the contractile unit of the cell. It's shown in this diagram here. And some of the key components of the sarcomere are labeled. We've actually gone on to tag many of these different elements in the sarcomere. I'll show you two examples here. Titin, which is shown on the left, and then MYL7, which is the myosin light chain 7, which was mentioned in an earlier talk today. So this is some examples of showing kind of what we can do with the live cell imaging when you're tagging endogenous proteins. So we've done this approach on over 30 different structures. They're listed here. Again, as Rick mentioned earlier, everything is open source, open science here, so all these can be found on our website. And we hope that people can use these lines. What we are using them for right now is to generate high replicate image data sets. So we've been doing this with all these lines here in stem cells and are starting to do this in cardiomyocytes to go towards our overall goal of building an integrated cell. So there's a few different ways that we can model this, and I'll just really quickly go over some of these approaches. So one of them is a statistical model, and in this way, because we have these high replicate image data sets at high resolution with different structures, we can combine them and use them as training sets for a model to help us predict where different structures are in the cell. And so what we found is that you can do this very well, actually, with just using the nucleus and the cell membrane as a reference point. And so playing here is an example of a cell that has predicted structures. These predictions are actually really quite good, so we're really excited about that. We've also now been able to translate this into a label-free approach, so we can do this without even needing a fluorescent protein. And again, the power of using these high replicate image data sets is that you can start to look at different things like this, just having starting with a bright field image and predict structures out. We can also look at different cells in parallel to kind of combine the raw data to look at how different structures can be integrated together to help us define what's going on in the cell state. So again, these models we're trying to use to help characterize and predict cell behavior. And so this is really what we've done at the structure level of the organelles. But as I alluded to, we're really interested in taking one step deeper and trying to understand what's underlying some of these organelle changes in the structural changes that we're seeing in the cell. So what happens if we add another layer of complexity? Multiple layers of measurements could inform the states of cells at individual inform the states of individual cells at a given point in time. So we focused so far on the organelle level, but we're interested now in the next stage to look deeper at the genomics level, and we're starting with transcriptomics for this. So again, just to kind of overview our concept for what we're trying to do with this is that we can look at cell level differences such as cell morphology, nucleus shape, and also cell membrane shape, in live cell imaging. We are generating the structure tagged lines so that we can look at structural organization in different cells and try to understand how cell morphology changes with cell structure and back and forth. But again, now we're looking at trying to see how the transcript level differences can help inform the cells at these different states. So our approach to do this is by doing single cell RNA sequencing and RNA fish. So what are some of the reasons? Why would we try to integrate at the structure level and the transcript level? Some of the questions we want to be able to ask with this are, does cell morphology or structure co-vary with gene expression? And if it does, can we predict gene expression given a particular cell morphology? And vice versa, can we predict cell morphology given what we know about the transcriptional state of a cell at that point in time? If we can do this, then this would also give us a way to link our high replicate imaging data set to transcriptional information such as an RNA-seq data set. So one of the first things we had to do to start this project is better understand the transcriptional states that our cells are in. And we did this by performing single-cell RNA sequencing. The experiment that we started with is looking at cardiomyocyte differentiation. And so to do this, we perform sequencing on 4 different cell groups, iPS cells, so these are day 0 induced pluripotent cells, induced pluripotent stem cells, day 12 cardiomyocytes, and then 2 other later time points of differentiated cardiomyocytes. We use the split-seq method, which is a combinatorial pooled combinatorial barcoding method of doing sequencing for single cells. It was developed in Georg Selig's lab at UW. And by doing this, we can sequence individual cells and cluster them in an unsupervised way. And so in this kind of a plot called a t-SNE plot, each dot here represents a single individual cell, and it's clustered in an unsupervised way based on gene expression relative to the other cells in that population. So with these plots, you can overlay different kinds of colors, either gene expression or different information we know about the cells. And so when we color code them by collection time point, we see that, not surprisingly, they tend to separate out, namely by the time point that they are collected in. So we have the stem cells up here, and then the differentiated cardiomyocytes that progress along from the earlier time points and then to the later time points. So we know that there is a large range of different transcriptional states that the cells can have. And so to just point out some of the obvious ones, we know that we have different cell types in this population. So first cell type I'll show is pluripotent stem cells. And so in these plots, the genes are color-coded based on the gene expression. So SOX2 cells are showing up red. And we see that that nicely labels the pluripotent stem cell cluster here. We also know that most of the cells that we put into this experiment were cardiomyocytes, and we can see that very nicely by looking at expression of TNNT2, which is a cardiomyocyte gene. And you can see here that, again, most of the cells are cardiomyocytes. So this is an example of cell states as 2 different cell types, but we can also look at, within one cell type, what are the different transcriptional states that cells could have. So an easy example of a way to look at this is by something that is known to change as cells age and further differentiate. So a classic example of this in cardiomyocytes is the expression of myosin heavy chain. So MYH6 is expressed at the earlier time points, and MYH7 is expressed later in development. And indeed, we can actually see that show up in our data where the earlier time points here, day 12 and 24, have expression of MYH6, and the later cardiomyocyte time points here have more expression of MYH7. So it's perhaps a little bit easier to look at this in a violin plot. And so just for simplification, I'm going to show just the 4 main clusters from each time point, not everything. So TNNT2, let's look at this first. So not surprisingly, there's no expression of TNNT2 in the stem cells. This is good. And then in these plots, it's really showing the distribution of gene expression in those given clusters. And TNNT2 is expressed broadly across these differentiated time points. MYH6, which is expressed earlier on in differentiation, has high expression at day 12, and then you can see that the expression levels go down with age. This is contrasted with the expression of MYH7, which is expressed lowly early on and then goes up laterwards. So it's kind of an example of known genes that we know would change expression over time, and that this is one potential way to kind of classify what's going on transcriptionally in the cells. However, not every cell is the same within these clusters. We know that there's more of a continuum of expression, and that there's a distribution and a range of what different states these cells are in. So we're an imaging institute, and so we wanted to take a look at this visually. One way to do that is by doing in situ hybridization. And so we performed RNA fish on different differentiated time points to visualize this a little bit better. So in this case, I'm showing day 12 cardiomyocytes. And the same 3 genes are shown here with MYH6 in yellow, MYH7 in purple, and then TNT2. And hopefully what you can appreciate is that there are some cells that have preferential expression for something like the early isoform, and then also some that are later, and then kind of the spectrum of expression profiles in between. If you look at a later time point, as expected, there's a broad shift in expression of this later gene. But what we're really interested in is that there are cells within here that have kind of a joint expression of these two genes. And this is just an example of 3 genes. You can imagine if we're looking at different combinations of lots of different genes, we suddenly have a huge transcriptional space that the cells could be expressing in different combinations. And while it's great to look at it in this way and in the single cell data, what we really want to know is there a way that we could actually link these transcriptional differences back to structural changes that we're observing in the live imaging? And better yet, could we actually predict these different transcriptional states by looking at a live image of the cell? And if we could do that, that would maybe give us a broader space of trying to understand what's going on in the cells at a structural level and also a transcriptional level. So again, we can do live cell imaging to look at cell morphology differences. We can combine this, and we already are doing this, with structural organization of different structure tagged lines. This is, again, with live cell imaging. And now we can do look at transcript level differences with RNA sequencing and RNA fish. But what we think the real slam dunk would be is if there was a way to really combine these 2 things, right? Doing these predictions based on cell structure and organization might tell us what's going on in the transcriptional state and vice versa. So I'm going to show just some very preliminary data of something that I think helps demonstrate one of the ways that we can look at this and maybe some of the things that we could study if we do that. So I'm going to show a video here of cardiomyocytes that are plated. So this is live cells. This is a Z-stack, and so you can see that the cells are beating as we go up and down in Z, and we do imaging with a nuclear dye just to use as a reference point for us later. So of course, these cells have a tag structure, and so in this case, we're looking at TNNI1, which is a cardiomyocyte gene, and just to simplify this, I'm going to show just a single plane from that Z-stack. And you can see that there's a variety of most of the cells are expressing this TNNI1, but there's also a range in expression and also a range in structural organization, nuclear shape, and also cell shape. So what we're starting to do now is think about ways to link this with the transcriptional data. So we can take the same area that we imaged live, fix the cells, and then perform RNA fish on the same regions. So the first gene that we'll look at is NKX2.5. This was nicely brought up earlier too, so hopefully you're already familiar with this. It's a cardiomyocyte transcription factor. You can see that there's low expression, so here the transcripts are labeled with these dots, and so most of the cells that have this TNNI1 also have the cardiac transcription factor, which is expected. But now we can add on another layer of information. Let's look at myosin heavy chain 6, MYH6. There are certain cells that seem to have higher expression. So if you look at these two neighboring cells, there's a clear difference in the levels of expression between myosin 6 in these two cells. What we want to be able to know is, are they structurally different? I'm not sure. Would we look at, we could look at this particular structure. We could also look at other structures and try to tease out if there are actually differences that, what makes this cell different from its neighbor cell. We can add another layer of information, again with another gene, so MYH7, same gene we looked at on the previous slide. You can see that in this case, it's more broadly expressed. And so again, we're trying to understand if there are, there's clear transcriptional state differences here, but if we could link this back to the imaging, that would be very powerful. So another way of doing that is kind of looking at different levels of transcript expression, but there may also be something else interesting going on here. So I'm pointing out this cell, which hopefully now you can appreciate has lower level of TNNI expression. It's maybe a little bit less developed in terms of sarcomere, at least based on looking at this protein. But if you look at just the expression of MYH7, it looks more or less similar to a lot of the cells that are around it. We look at just that cell, zoomed in again. This is just showing the MYH7 transcript. And hopefully what you can appreciate is that it looks like there's actually some localization of the transcript that's aligning onto the sarcomeres here. So I think this is an example of something that, with just the live imaging, we can tell that the cell is different. If we looked at just the transcript, you can also see that there might be some localization differences compared to neighboring cells. But then when you combine the two of these things, you can really start to look at some interesting biology. So in this case, we think live cell imaging is providing information about the cell dynamics, the morphology, structure, and also cell neighborhood. And that RNA fish is providing spatial context for the gene expression of things that we're looking at by RNA sequencing and eventually, potentially some other methods. So to kind of summarize and highlight the overview of this project, I mentioned how we're using endogenous tagging in live cell imaging in pluripotent stem cells and also in cardiomyocytes, that we are generating high replicate imaging data and using machine learning to build predictive models and really trying to integrate these different structures that are tagged to understand the cell and look at it on a more structural systems level. And then now we're also starting to look at the omics and trying to combine single cell transcriptomics and spatial integration to try to integrate some of these things on different levels. So what we're hoping that we can do is start to predict transcriptional profiles based on just a subset of surveyed genes, like I showed you in the RNA fish. We also hope to be able to predict transcriptional states from cell morphology or from organelle structure, and also do the reverse. Look at cells that have a particular organelle structure and find out if we can actually predict what the transcriptional state of that being, where that cell would fall in that T-SNE plot when I was showing about the different kinds of gene expression overall. And of course, as an institute, we're continuously working on editing new structures, and we have a lot of efforts going towards making this integrated cell in the stem cells. We're starting to do this now with the cardiomyocytes with the same structures and also some cardiac-specific structures, as I showed before. So I'd like to thank everyone at the Allen Institute for Cell Science. This is a highly interdisciplinary project. We operate on a team science platform here, and so everything I presented kind of spans the work of multiple different groups and many people. So thank you for everyone for that. I'd like to thank you all for your attention, and I'm happy to take any questions.
Hi, cool. I was wondering if you could potentially add lineage on top of everything else that you're looking at. So like, say at an early time point, a cell that has maybe MYH6 and MYH7, if you can identify that live and then find out whether that makes something different later than cells expressing one?
Yeah, that's a really interesting question. At this point, we haven't. We are doing some time-lapse imaging, although we're not currently working on integrating that too much in with the predictive models, although I think it's definitely a really interesting direction. Another thing that we're working on that's kind of related to this is the early fate decisions that are happening during cardiomyocyte differentiation. And so we're starting to develop different methods of doing that high-resolution imaging as the cells are differentiating. So I think that there could be some really cool fate decision points if we can identify some key structural organization differences, and we do see those with certain lines early in differentiation, and then overlay that with some of the gene expression. I think that would be a great application.
Yes, hi. Do you plan to apply the same pipeline for tissue extracts, organs, like, I guess, using mouse transgenic lines instead of cell lines?
At this point, no. We think that, we chose to use induced pluripotent stem cells because we can differentiate them into many cell types. It's also clearly a very complex system, and I think that what we've been finding is that the stem cell model is a very good model for doing those kinds of things. We're hoping that the basic tools and kind of trying to understand the ground rules for the cell, that those will apply in vitro and also hopefully be able to be applied to other people who are studying different tissue or disease models in that way.
Hey, Kaytlyn. That was really great work. I was wondering, have you guys teased out gene expression in cardiomyocytes that are showing obvious signs of maturation, such as binucleation or something like that?
That's a great question. Not yet. We're definitely interested in looking at some of those things. So I think with the binucleation is something that we see. There's a lot of even just like nuclear differences in terms of the size of the nucleus, the shape of the nucleus. That's something that we're starting to look at closer in the stem cell model. But now as we're developing the tools to do this in cardiomyocytes, I think that could be a good way to look at it.
I'm curious about how the data from the t-SNE plots compare with the data from the fish. It seemed like you had two different myosins, which were kind of, I think, day 24 versus day 90 or something. And yeah, you had quite a lot of cells that seemed to express both by fish, and yet those two populations were really separated. Like there was no transition between day 24 and day 90, which was kind of amazing.
Yep. So the comment I'll make on that is that there's a lot of heterogeneity, and that I think is one reason why we might be able to look at this stuff with a lot of high replicate imaging within given time point. And I think within a given time point, there's definitely an overall shift, and yet we still haven't characterized this stuff yet. This is kind of brand new work, but that's something that we want to analyze. I think that we've also biological context, there's variability in differentiation, the different cell types and stuff, and so I think that's something that we'll definitely look at. But yeah, it's interesting that in some cases there's kind of a clear switch in expression. It seems like things are pretty well correlated with what we see in the t-SNE when you look kind of at a broad level, but I think we'll start to probably pick up differences when we look really deeply at particular genes.